Hi, everyone. Welcome to today's Micro Minute. My name is 𝗶𝗶𝗃. I'm a senior application scientist here at the ZEISS Microscopy Customer Center in Dublin, California. I'm really excited to talk to you guys today about CAR T cells and specifically how I think the Lattice Lightsheet 7 is a really great way to image this application space. And without any further ado, I think we'll dive right in. I've got a little bit of something for the biologists and a little bit of something for the microscopy enthusiasts. So hopefully everybody will learn a little bit of something today. So you may or may not know this, but right now there is a chance that your body could actually be actively fighting cancer cells or cells that have divided and have some DNA damage and are really no longer the tissue that they're supposed to be. The way that this happens is your cell basically makes a bad copy of itself, and your immune system actually recognizes that as no longer you, and they will go in and kill it. So your T cells, part of your innate immune system, will recognize that and get rid of those cells. Unfortunately, of course, that doesn't always catch everything, and we probably all know somebody who has had cancer, and what can happen if your immune system doesn't catch these cells in time as they rapidly proliferate, or perhaps they've disguised themselves very effectively, is those will continue to divide and turn into a tumor and eventually start metastasizing in other parts of your body. And we're all pretty familiar, I think, with the fact, because most of us at this point have known somebody that has been affected by cancer, typically we treat that with chemotherapies or even with radiation treatment. And unfortunately, that has a lot of side effects, right? Those are not very targeted therapies at this point in time. And so you sort of have some friendly fire. And, if you know somebody who's gone through chemo, they typically lose their hair. Your skin is very sensitive. Your gut can be very inflamed. And that is because you are targeting all rapidly dividing cells rather than specifically going in and saying, I just want to kill and root out these bad cells that have made bad copies of themselves, essentially. And so what I want to talk about today is how there are these emerging engineered therapies that basically harness the power that your body has to take those immune cells, those T cells, and we want to re-engineer them to now go out and root out those specific cancer cells. Because now we have a much better understanding of the fact that certain cancers have very specific markers on the outside of themselves, right? You might have like a HER2 positive breast cancer, for example. We know that specific cancer diseases will have very specific antigens on the outside of those cells that we can then engineer immune cells to then go and specifically target, right? So now we're not going to kill all of our healthy, rapidly dividing cells in addition to the cancer. We will just target those cancer cells. And I'll talk a little bit more about that. Right, so this is a really burgeoning field. A lot of really exciting research is coming out on this, and just you can see there's a lot of review papers coming out in the last couple years. So it's a super exciting field, so that we don't have to deal with all of these awful side effects when we are targeting a cancer treatment. And you might be thinking, Rebecca, what on earth does this have to do with the Lattice Lightsheet? So I'll get into that. What I want to highlight today is some really cool experiments that I got to do when I was doing Lattice Lightsheet workshops. And I visited Memorial Sloan Kettering Cancer Center in New York. And specifically, the Huse lab sent somebody to spend some time with me on the instrument. So Elisa Sanchez brought me some really cool samples, and she was having some challenges with live imaging, right? So if we're thinking about generating these CAR T cell therapies, you have to think about, how are we validating this? How are we going to look live and see, as I add T cells, are they going to kill the exact targeted cancer cell? All of that stuff. This lab looks at how the immune system interacts with different cell types. Specifically, she's interested, at least at the time, her research was focused on how cancer cells act in cytoskeleton, is reacting from the T cell attacking it. But again, you can extrapolate that to this burgeoning field of CAR T cell therapy, and how validating that on light microscopy systems is a really effective way to understand what is going on at the cellular and subcellular level here. And so she brought me this experiment, and we had some really exciting results. She was having challenges on other fluorescence imaging systems because, think about this, right? The experiment setup is you have a plate of cells. plate of cancer cells, in this case melanoma cells that are labeled in green here for the actin cytoskeleton, and you have to go in and add your T cells, and these are just stained with cell view maroon. So if you are doing CAR T cell therapy, you just add stain and you can image these in a dish immediately. But she was having the challenge of how do you know which cancer cell is actually going to be attacked by a T cell? And so her field of view is pretty restricted. If you do have a lot of experience imaging at these resolutions on a light microscope, whether you're at 40x or 63x, your field of view is going to be pretty limited typically, right? Like on a 63x, you're going to be less than 100 microns in X and Y. And so not only are you having to hope that you pick the right cell that actually gets attacked during this experiment because you don't know, you're going to have to hope that it happens in your field of view. And so she brought me this because she was having a lot of trouble. I think they might have had a spinning disk system. And I'll just pose a question because we do have a live chat here if you want to ask questions or answer live. How long do you think it took us? This is a multi-position experiment. How long do you think during this workshop it took us to get this done? We imaged every 3 minutes for about 2.5 hours in these time lapses that I'm showing you in multiple positions. Do you think it took us like a whole week? Did it take us 4 days? It only took us our half-day session. She had one time slot with me, and we threw this sample on the instrument. We tested the initial conditions to make sure I had the, excitation settings set up right. And then after that, we just set up multi-positions and imaged this for 2.5 hours, and we got this in our first attempt. That is how easy it is to use the Lattice Lightsheet from ZEISS. And so I want to talk a little bit more about that in a second. But again, this is a really exciting data set. You can extrapolate pretty easily how well this would apply if you are doing CAR T cell therapy research. Now you can go in and see, is it actually attacking your antigen of interest, the cells that you have engineered? What is happening? Again, in this case, we're interested in how the cytoskeleton of those cancer cells is depolarizing after getting attacked. There's all sorts of questions that you could ask that you probably couldn't ask before. These are max intensity projections, so this looks like it's just a 2D data set. It's not. It's a 3D data set, right? And so let me open that up, actually, so you can see more closely. Okay, so this is one of those positions in Zen, and you can see I've got this lovely bright green melanoma cell, and we can click through the time series here until we see, look, we even see some T cells float by, right? They don't even care about attacking this guy until we get a little bit later in the time series. And now we can watch this come in here and actually start to attack the cancer cell in 3D. And, you can go in the software and actually use these clipping plane features and look more closely. And I've actually linked down in the description below a video that one of our other application specialists did explaining how we can use a 𝗶𝗳 to actually track what is going on in these videos. Let me just bump this up so you guys can see it a little bit better when we have the clipping plane on. Okay. So that is linked below, and you can see, because once we get the images, that is like step one for you guys. We really need to actually do some analysis after the fact. So we show some proof of concept work of what that looks like. But we can really see what's going on in 3 dimensions here. So this opens a whole new world of research questions that you probably couldn't think about asking before, because we're usually limited when we're doing these live fluorescence experiments, because we're going to be worried about phototoxicity and photo bleaching and how fast can I image to get this volume while things are happening, right? So a lot of challenges that typically happen on a standard inverted system that the Lattice Lightsheet works around. So how does that work? I promised you guys that I would go into some of the microscopy as well. So this is the application space, but how does this all actually work? And let me go back here. Okay, so for those of you that are used to working on a standard inverted system where you're putting your 35mm dish or your 96 well plate on, that's what I'm going to refer to as an epi-illumination system, or that's like your spinning disk or your laser scanning confocal. And the way that works, I have a zebrafish for our example here. Of course, we're talking about cells on glass. We're going to go in for our optical section, which I have represented in green here, and we're going to focus on that section, and we're going to scan through, and we're going to throw away the out of focus stuff, but we're going to have illuminated that entire zebrafish in this case, right? So now imagine if you have to take 20 slices to get your Z volume, or 50 slices. Now you're, for one time point, illuminating that entire sample 50 times, for example. And then for this experiment that we did about 50 time points, so that every 3 minutes and 2.5 hours, that's like 50 stacks in Z times 50 time points. That's a lot of light being put into your sample. So the way that light sheet microscopy can be a lot gentler than standard epifluorescence microscopy is we're going to take that detection objective that you see here, and instead of illuminating through that objective, we're going to have illumination objectives perpendicular to our detection optic. And so now we're able to optically section only in the region that we are focusing on, right? So we've only illuminated that single plane. So now, if you are taking, let's say, 50 different planes, you're only illuminating those planes when you're focused on them, right? So now you can see how that is an order of magnitude gentler than a standard epifluorescence system. And you might be thinking to yourself, but Rebecca, how on earth will that work if I have a dish of cells and I'm trying to illuminate into the glass? That sounds like a nightmare in terms of physics, right? And it would be. But that is not actually how a lattice light sheet works. Instead of suspending something in a chamber like we do on a classic light sheet with cleared brains or with zebrafish, whatever your sample is, we do just have our cells in a dish. Pick your favorite number 1.5 cover glass bottom dish. And instead of having to try to image through that, our optics are rotated, right? So the initial configuration of this had a dipping lens system, but the ZEISS implementation it's going to feel just like a regular inverted microscope that you're used to bringing your growing cell samples to. And so we have the illumination optic 30 degrees to the bottom and the detection optic 60 degrees, so they are still perpendicular to each other, and now we can scan our sample through that. And that is how we were able to do this multi-well T-cell experiment. So, right, orders of magnitude gentler than standard epi-illumination. I just alluded to the fact, I kind of buried the lead here, but there are different flavors of light sheet microscopy, right? And so what a lot of people are used to thinking of is this classic light sheet, this Gaussian light sheet. And that is a scanned Gaussian beam that, again, it has all these wonderful benefits I just talked about. You can have much less bleaching, and you're much gentler on your samples, but that beam is thicker than we would want to be able to resolve cellular structures. Because in light sheet microscopy, your optical section or your resolution in Z is dictated by how thick that sheet of light is, because that is how we're optically sectioning. What you would need to resolve subcellular structures is a thinner light sheet, like a Bessel beam, but the challenge with the Bessel beam is that you can see in this illustration here in the middle that it has these things called side lobes. And so while it's a thinner beam and you would be able to resolve smaller structures, it is going to put more light into the sample, which is what we're trying to avoid, right? And you're also going to have to deconvolve that out of focus light out of your images. What Eric Betzig discovered is that if you take a bunch of these Bessel beams and you line them up perfectly beside each other, you can use physics to your advantage and you get destructive interference of all of those side lobes, and now you have a very thin lattice light sheet. That's where the name comes from. You have a sheet that looks like a little lattice structure. And all we have to do now to make it a uniform sheet for imaging, thin enough to resolve subcellular structures, is dither it really quickly back and forth. So just move it with a mirror back and forth very quickly. And what we're able to do with that are experiments like the one I showed you earlier, or like this example here, where we have cells imaged continuously for 9 hours in this example. That's 1 million images in total. Now try to get 1 million images or that many time points on your standard epifluorescence system. You're gonna bleach or damage your cells very quickly, and your cells are gonna have a bad time. They're not gonna be ruffling at the end of their video. So the way that this works is, I mentioned it feels like using a standard inverted microscope, and that is because rather than the initial configuration, like I said, which is a home-built dipping lens system, which for those of us coming from cell culture world, that's not the best system in terms of preventing contamination or having a good, nice incubation setup. And so when they were engineering this to be a turnkey system from ZEISS, we said we really need this to be an inverted setup. You want to be able to take the glass that you're used to culturing your cells in, or a 𝗦ci𝗰𝗶𝗹𝘀 plate, or a chamber slide and just put that on the microscope and image just like you normally would. The way that the magic is able to happen, because now you're trying to take this very complex physics, right? I was just talking about destructive interference and perfectly shaping a beam to make it the right size. You have to go through air and then glass and then water and then glass, right? There's a lot of different changes in refractive index. And but don't worry, you don't have to think about that because we have a meniscus lens. That is how all of this is possible in an inverted setup. I mentioned we would have stuff for the microscopy enthusiasts, so bear with me if you were just here for the biology. But that is how it works, and if you have questions about it, feel free, again, to drop it in the chat. We also have some built-in precision optics because no two cover glasses are manufactured the same. And so we do have to correct for that change in cover glass thickness, but it's just a very quick thing in the software that I will show you guys in a second. So again, I bring all this up because, right, Eliza brought her sample to me. We only had half a day to try to get this data, and it is that easy to use that it's a sample that I had never seen before, I had never worked with before, and we could get it done in a single shot because, well, one, she brought me beautifully stained samples, but two, because it is that easy to use. It's just like bring your sample to an inverted microscope. It feels just like using one. It's super easy. You don't have to worry about all of the intricate alignments and things. You don't need to be an optical engineer. So with that, we're going to go visit me in the past, get in our time machine, and we're going to take a quick look at the Lattice Lightsheet. So you can see here, this is a super compact instrument. It looks like a little printer, but it's really amazing. The optics that are built into this are very powerful, and they've really engineered it so that it's super easy to use. If you have ever seen the home built configuration, it looks something more like this, right? It's quite large, takes up a ton of space. And again, here you don't have your incubation to keep your cells going for days at a time. You really have to dip these in the system, the objectives, dip them into your sample, excuse me, and you have to be able to align the mirrors and stuff. It's not super user-friendly versus what we have now, which is this very compact, easy-to-use system. And I'll go into a little bit more of the features that we built in here, which again, you can just bring me your sample. Please reach out to us at the ZMCC if this is an application space for you, and we will be happy to show you how easy it is to get the data that you might be struggling to capture right now. So with that, let's take a closer look at the hardware because it's got some really cool stuff. I like to call the stage my little Mars rover because it is a 5-axis stage. So for any of you who are used to having to manually level a stage, like using the screws, leveling screws, and doing all of that, you don't have to worry about that here. The stage moves in X, Y, and Z via piezos, which is how we're able to capture those really fast volumes, up to 3 volumes per second. And you can also tilt in X and tilt in Y, but you control that in the software. So I'll show you how we control that in the software in a second here, but that's why it's so easy to get everything nice and level. Again, we are dealing with pretty complex optical physics here to make this possible in an inverted setup, but we've made it very easy to use. And some of that is these features here, like I'm showing you the tilt of the stage in X and Y, all controlled in the software. So leveling is no longer a nightmare. It's super easy to do. Because it is necessary to do. And then, of course, this is a water immersion system. We have a 40x 1.1 NA objective on here, but you don't have to worry about your samples drying out. It has auto immersion built in via the software, so it will keep refreshing your sample overnight for your long time lapses. And that is built in here. I'm going to lower the front in a second so you can see that water bottle in the front where we keep our immersion water. And that is just piped into that meniscus lens that I was just talking about to make sure that very easily you can keep that interface wet for the entirety of your experiment. This transmitted light arm lifts up. You might notice there are no eyepieces. Fear not, we really don't need those anymore. But this transmitted light arm you can image in bright field with white light, with red LEDs, or with side LEDs, depending on what you need. So that way you can find your samples and then switch over to fluorescence. You can also just take overviews in fluorescence if you need to make a little Google Maps of your sample that way. And then the incubation setup is one of my favorite things. It's very sleek and relevant for this. We have these 2 ports. So if you do have a microfluidic setup, maybe you're adding your T cells with microfluidics because you're really fancy. Maybe you need to add drugs and wash out drugs. We have this set up so that you can very easily add that to your sample during the experiment, and then it clicks in with these pins. So it'll tell you when the lid is not connected so you don't start your experiment without the lid on. Helpful for people like me who sometimes are forgetful. And then, of course, that's all heated. And then, of course, you can control for CO2 and humidity. And we even engineered it to control for oxygen for those of you that are doing any sort of iPSC work and have sensitive stem cells. Of course, for this CAR T-cell work, you really typically just need the 5% CO2. But very, very sleek, compact design. It can fit in the corner of your lab very easily. And again, super easy to use. Most of the stuff you're going to do is through the software. So let's jump into that. I alluded to the fact that there is no more nightmare tweaking of leveling screws. You literally just look at the reflection in the software and control tilt in X and Y that way. So it's super easy to do that. That's just the first thing you do. You plop your sample on, level it. So if you are coming from a home-built system, this part is a dream in comparison. And then after you level it, then we can just focus the beam. So I just want to walk you through how easy that is because, again, we were able to image ELISA sample in like one shot. So we built in a webcam into that where that LED is, so you can take an overview of, in this case, a chamber slide, find where you are in your sample. That's why we don't really need the eyepieces. And from here, you can start to make your multiple position experiments, make a region of interest. And to focus the beam, it is maybe one additional step compared to what you might be used to on a typical inverted system, unless you work with SIM, in which case this first step actually will feel very familiar. The first thing we're going to do is turn the dithering off, so you want to actually see that lattice structure on your sample, and then we're just moving the beam. You'll see an animation in a second where we're going to move that sheet relative to the back optic. That's the first focusing step. We're just looking for a sharp grade. If you do SIM imaging, that's going to feel very familiar to you. The next thing we do, we don't actually move any of the hardware because, again, if you've had any experience with a home-built system, there's so much alignment that goes painstakingly involved in setting up this complex optics. We don't have to worry about that. Everything stays set, and then we just move the beam relative to the hardware, so the hardware is stationary. It takes a lot of the headache out of it. So here's where we move that sheet waste, the thinnest part of the light sheet, into our sample. That's going to feel like focusing a regular inverted system, just that instead of moving the objective, we're moving the sheet. And the very last step, we just control for that cover glass thickness. So we control those phase plates in the detection optic, which again helps us correct for the slight differences in thickness in number 1.5 cover glass. But that is how easy it is. You just hop on 1, 2, 3, and now we're ready to image. You can start tiling, start doing multiple positions, just start imaging in your single spot, whatever you want to do. And that's why it's so easy to just, again, hopefully your staining is good, you know. You want that part optimized first, but once you have a really good protocol for getting good fluorescence images, it is that easy to just knock out something complex like this that you might be struggling with on another system, and knock it out even if you only had like a half a day to try this out, right? And so that's why I wanted to bring you guys this story today. And again, I've highlighted Elisa Sanchez's paper related to this type of work in the link at the bottom, in the description at the bottom, as well as a link to the Huse lab, who have been so gracious to let us share this work, as well as, again, the 𝗶𝗳 tracking example that we've done with this experiment. With that, if anybody has any questions, again, I'll give you guys a second to add them to the chat. I know since we're live, there will be a little bit of a delay. But while I'm waiting to see if there are any questions, please reach out to us separately if you're not watching it live. But we also run workshops regularly. So at the ZMCC, we have light, electron, and X-ray microscopes, and we always do virtual and in-person workshops to help you guys make sure that you're trained and basically running the system to the best of your ability and not missing out on the wonderful technology you have in your hands. So those are on a rolling basis. You can find that here. Of course, we always have the portal. That is where if you are just a little bit more, you want to learn it on your own, you're feeling pretty proficient. If you have a ZEISS microscope, you can always go there and cheat yourself. We have a lot of wonderful training content there. And then, of course, our upcoming live streams. I believe next week we have an electron microscope workflow. And, of course, all of our past live streams are there as well. You know, we really want you guys to know that as our customers, we are here for you to come and pop on with us live and ask us your most pressing questions and hopefully learn something new that you didn't know about before. So with that, I'll give it another second, but thank you so much for your time and attention. And again, if you have exciting CAR T-cell work that you're working on. Would love to image your samples here at the ZMCC, or any other cool stuff. Just I'm always begging for cool samples. We love to see what our scientists are working on out in the field. And with that, have a wonderful day.